out of all the steps, this is the most important step that is asked in the exam. Restriction endonucleases. Now, what are they? They're actually enzymes. So these are enzymes. And what they do is that you have particular sequence of bases. So let's say you have a gene. Let's talk about our insulin gene. You have an insulin gene. Before the insulin gene and after the insulin gene, there are particular sequence of bases. And these sequence of bases are called recognition sequence. So this enzyme is going to attach to these recognition sequence and is going to cut the DNA. So this is what it does. You have to know this definition for exam. So restriction endonucleases recognizes and cuts DNA. Number one. Where? At specific sequence of base. These specific sequence of base are called recognition sequence, and which has a palindromic sequence. That I will explain later on what that means. So we have so many different types of these enzymes, and thankfully you don't need to know any of them. These are different examples, and I'm showing you here, like these ones, these enzymes where they cut. So these are the sequence that is going to recognize and cut, recognize and cut. Okay, so let me give you an example here then. So this is my DNA, and let's say I want to cut here. So the one highlighted in yellow is the area that I want to make an incision, I want to cut. So the enzyme is going to basically attach to this area and is going to cut it. Now, the way it cuts is very, very unique. It does not do a blunt cut, but rather it cuts uneven, so it cuts in a staggered fashion. So as you can see here, this is before and this is after the cut. Look at how it cuts. It's in a staggered fashion, right? There are this leaves uneven cuts. So each strand here, this strand has a unpaired base, exposed area, and this also has an unpaired exposed area, and we call them sticky ends. We call them sticky ends. So this enzyme, restriction endonucleases, it cuts at recognition sites and it leaves sticky ends. Now, what is palindrome? Palindrome is basically, if you can see the sequence here, look at the sequence here. So this whole thing is called recognition sequence, but they also have something very unique. It's called palindrome. Palindrome, if you see here, A, G, C, T, T. Now, if you go to this side here, it's also A, G, C, T, T. It's anti-parallel. It's the same base in the opposite direction, and that is called palindrome. So these recognition sites are all have palindromic sequence. So A, G, C, T, T is A, G, C, T, T. It's the same sequence, but in the opposite direction. Now, you're thinking, why on earth are we cutting it on sticky ends? Now, it makes sense why to cut it, right? Because we want to get the insulin gene out. So why do we cut it in a staggered fashion, with sticky ends? This is what happens, okay? This is let's say this is our DNA. So this is our DNA, and we cut the DNA in one part of it, this one in a sticky end, and this is our our vector, our plasmid. Obviously, we want to join them together, so this is how it's done. So the vector and gene, both of them are cut. So these are 3 points in the exam you need to mention word by word. So vector could be the plasmid, and our gene, any gene, they are cut with the same, very important, same enzyme, same restriction endonucleases, because we have to make sure they are complementary to each other. So therefore, they both have complementary sticky ends. So when you join them together to form your recombinant, they have to join together by what? Complementary base pairing. And they are mixed together and they are joined together by using the enzyme ligase. This is another one showing you the overall steps. As you can see here, our DNA has sticky ends, plasmid has sticky ends, and DNA ligase will join them together. And this is also another one showing you same thing, sticky ends, and then join together.